Hello. So we learned about neuroplasticity in chapter 8, but I'm going to briefly recap before I tie in my specific area of interest with regards to this. So we all know that neuroplasticity refers to the brain essentially rewiring itself to increase the efficiency based off of the feedback that it's receiving. And we covered the depths of the changes when we discussed LTP and LTD. So with regards to potentiation, we know that an axon terminal frequently releasing neurotransmitter into the synaptic cleft and frequently stimulating the target cell by lots of neurotransmitter binding to its receptors on the postsynaptic membrane will result in a larger response in the cell. So potentiation can result in more neurotransmitter released with each action potential, an increase in the number of neurotransmitter receptors in the postsynaptic membrane as kind of seen here, changes to the types of neurotransmitter, so that's kind of seen here a little bit, or responses that occur through second messengers increasing their sensitivity to neurotransmitter and leading to a bigger response in the target cell. So, and that's kind of seen here. So depression, on the other hand, synaptic depression is, that's shown here, and that's essentially the opposite. So it's occasional action potential reaching axon terminals, and it results in a decreased response. So the strength of information flowing through the nervous system can decrease parts of the neurons, and chains of neurons that we rarely use grow weaker. So we know the amount of neuroplasticity is highest during development, and it's present through life, just in a lesser amount. So I am interested in its transient increase and importance in recovery from traumatic injury. So basically when it's trying to rewire itself back together after it's been injured. So traumatic brain injury, or TBI, basically this slide is just going to show the effect of TBI and how it affects at a molecular and anatomical neuronal change level. So I pulled this image from the journal article Traumatic Brain Injury and Neuronal Functionality Changes in Sensory Cortex, and that is listed in my references at the end of all of these slides. So I chose to include it because I think that it adequately highlights the evolution of neuronal injury at varying levels and time frames. So as you can see, traumatic brain injury includes molecular anatomical changes as well as changes in neuronal functioning and processing, and this is just kind of breaking down that process and the duration. So we can see axons experience tears, swelling, and disconnection. And also we see TBI causes activation of molecular cascades, and those cause the secondary brain damage via the multifactorial processes, including oxidative stress and excitotoxicity. And that's kind of shown down in this part down here. So this slide, BDNF and curcumin. So this is going to be a breakdown of the components and why they're significant in relation to TBI. So we'll start with BDNF, which is over here. And you can probably make the connection between TBI and BDNF based off of what we've learned previously. BDNF is synthesized predominantly by neurons located in the hippocampus, which we know is associated with processing cognitive function, and it's also relevant in the induction of LTP. So BDNF can also facilitate in synaptic transmission and regulate gene expression through activation of synapsin and Krebs. Curcumin, which is shown over here, on the other hand, you may not be familiar with. So curcumin is the bioactive substance in turmeric. And it's widely known for its anti-inflammatory properties, as well as studied in disorders with oxidative damage or cognitive deficits associated with aging, also in the case of Alzheimer's, with its amyloid pathology. Okay, so now that you have a refresher of several familiar terms and processes that we've covered in class thus far, I'd like to correlate that information with a journal article that tied these components and used the foundation for what we know about these elements to evaluate a unique and potential treatment method for traumatic brain injury. So, in a journal article published in Experimental Neurology, Wu and his colleagues evaluated curcumin effects on cognition and neuroplasticity. So this slide is a high-level overview of that study, showing basically here TBI leads to these free radicals that are created in response to it, which in turn reduce the levels and function of the BDNF and its synaptic plasticity effectors, synapsin 1 and Krebs. So these changes may underlie synaptic dysfunction, which you can see here, as well as cognitive impairment observed after TBI. So the study measures curcumin and it's as a method to counteract these effects here. So the exacerbation of TBI, you can see this HF here. The exacerbation of TBI effects in the presence of a high-fat diet were also measured, but my focus will be predominantly on the effects of BDNF system and its downstream synaptic plasticity effectors, synapsin 1 and Krebs. Okay, so the results. Curcumin proved experimentally to be sufficient in the reduction of oxidative stress impact on the BDNF system, ultimately suggesting that it may be an effective therapy to counteract the effects of TBI on neuronal plasticity and functioning. So breakdown with regards to this slide here, as far as terminology goes, this FPI here refers to fluid percussion injury. This CUR here is the curcumin. The RD is a regular diet, like I kind of mentioned in the previous slide. They also looked at, they analyzed a high-fat diet. The sham that you'll see here is basically just their control. And basically what this is showing, the results of the study, curcumin in the diet normalized BDNF levels in the hippocampus of TBI rats that were fed a regular diet, which you can see here, and that's at about 97%. And then also, less importantly, in the high-fat diet, it also had an effect there as well. So my references here. So the imaging was pulled from Frontiers in Neuroscience and the cellular scales, and that is the, as you can see, the imaging I used for BDNF and also LTP and the neuronal effects of TBI. All of those details I obtained from Karen here, this article. And the study that I just referenced is from this site here from Wu and his colleagues.